Hello, I'm Katie, your biology 110 instructor. So tonight we're going to be doing the diffusion and osmosis labs, and we're not going to do the whole part of it tonight because the eggs need to be deshelled, but there is a lot of prep in this lab, so I thought that would be helpful. Okay, that's actually a whole lot of the lab. So we may do the potato part, but we're definitely going to make the solutions. We're not going to do the eggs tonight because we have to deshell them. So let's see. The first thing you need to do is make sure that you have your diffusion and osmosis lab prep, or your diffusion and osmosis handout is what this is. And it has the lab preparation. So you need to make sure that you have all of these things. And this is why we're going to need to take extra time tonight, is because you need to make these two of these solutions, and you need to get the eggs ready. But we may end up doing some of this other stuff. You just save the eggs for later. The other thing you have is your answer sheet. And here's the answer sheet. And this is the part you're going to submit for a grade. And notice you need to watch this video, and then you need to answer the questions. And you need to watch this video and answer the questions. These essentially are the activities that we're doing with maybe a few minor differences. So make sure that you watch those and understand them. And here is another one. And we're doing this experiment as well. And you can have either celery or Napa cabbage, or I may try romaine tonight. So make sure you have that. And here are some questions that you might get off of your lab packet. And then your results. So you're going to need to make sure that you record your results here, and your osmosis and your potato data, and the 3 different solutions, and then osmosis and your egg data, and so forth. So the first thing we're going to do is, let's see, stop share, is we're going to make sure that we have everything that we need, and I do, except for the deshelled chicken eggs. Then the 6 clear cups from the lab kits. I even have some tapes to label them, so I have everything here and there. So, oh, I don't have saran wrap, but I can go get that. So for our solutions, that's what we're going to make tonight. And what I brought were some empty water bottles that I can make them in. And we're just going to need 2, but then we'll need a third, which is distilled water. And I'm actually going to use, it says that you can use tap water or other water for that. So I'm going to go with tap or purified. Distilled is better, but I'm using the same water to make all the solutions, and it is purified. So I'm going to go ahead and use the same water. Make sure that you label everything. And here we go. So I don't know what's going on with my camera, that it's turning on and off tonight, but these bottles are so lightweight right now, and they're almost squishy, and they'll stand up a little better once they have everything on it. But here is, it's not distilled water though, but I just labeled it D2O because it's purified water, and that's the water we're using tonight. Okay, so we need a 300 milliliters of a 15% salt solution. This is going to be our hypertonic solution, and that means that you're going to have more solute in the solution than you would have in the actual cell. So you can get a weigh boat or a Dixie cup and turn on your scale and make sure that it zeroes out. And Well, you can't tell on there. It looks like it has 8, but to me, it looks like it has a bunch of zeros. So then we're going to take our salt, and we want 45 grams of salt to 250 milliliters of warm water. And here, we'll just tap water also for this. This is going to be a lot of salt. And let's see. $21. My salt is already pumped up. Up. Got 22. Oh, 45. And 45. Okay. Now, my water is not warm right now. So what I'll probably do is take this water, or take my solution and pour it out after we're done, put it into a bowl and heat it up a little in the microwave, and then until this dissolves, and then let it come back, to cool, and then put it back in. So this right here is our 45 grams of salt and to 250 milliliters of warm water. And then we have, let's see. There's 50. So let's not let me lose count. That's 50. Oh, 100. 150. 200. 'And there's 250. Okay, now here's the thing. You have to figure out, you have to end up with 300 milliliters of solution, okay? And we can't tell right now how much there is in here because of all the salt. And by the way, this is going to, I'm going to go ahead and do this. It's still just such a flimsy bottle. It's not easy to put this tape on. But I'm going to go ahead and put it on anyway. And I thought maybe putting some water in would help it a little bit. And this is going to be our 15% NaCl. So we know we have somewhere between 200 and 300 milliliters. And this is actually dissolving better than I thought it would. So I may not have to get it too warm. But I have no idea how much of the solution is in here because of the salt. So I'm just going to check and make sure it's still good. And we've got, now really this would be much more ideal with 1,000 milliliter, that's 50, 1,000 1,000 milliliter graduated cylinder, and you could do it all in one fell swoop. And you could just fill it up with a little water. It wouldn't be as complicated as this. That's 100. Yep. $150, $200, $250, well, that wasn't good because I spelt a little bit, which is going to make it a little bit off. Still going to be a lot more hypertonic than before. So this is going to be up to 300. So we're going to have to fill this up to the 50 ml line. Okay. And then we're going to go, there we go. So that's 300 now. And I'm just going to go ahead and look, none of the salt is settling on the bottom. That's good. So it's warm enough. I'm going to go ahead and pour this all back in here. So this is our hypertonic 15% NaCl is made already. Okay. So we have our NaCl and we have our distilled water for our 15% NaCl. Now we need an isotonic solution, okay? So basically, let's see, a hypertonic solution has more solute or dissolved materials in the solution than in the inside of the cell. So this has a lot of salt in the 15% in it, and it's going to end up drawing the water out of the cell because water always moves, anything always moves from high concentration to lower concentration unless there's active transport. But this is just osmosis. And if you all remember back from Mod 1 in the background material, osmosis is diffusion of water through a selectively permeable membrane. So salt's not going to be able to get through the water because it's hydrophilic and it can't get past the phospholipid bilayer, which is hydrophobic. So the salt's not going to be able to move. So the water's going to be moving, and water's going to go from high water concentration in the cell to lower water concentration in here in the 15% salt. So anything you put into this solution should shrivel up any living cell or something like that. Because... Okay, so this is hypotonic, our distilled water. This is going to have a lower solute concentration than just about anything that you put it in, okay? Any of the cells that you put it in, because this doesn't have any solutes, okay? It's just plain water. So water is always going to move from this solution to the inside of the cell, okay? From high water concentration to lower water concentration. So this will move inside the cell and it will make the cell swell up. So if you ever like put vegetables in your fridge and you put them maybe in water to keep them crisp, then that's why they're crisp, because water's moving in. Or if you have a really wilted plant and you put water, you water it, then the water's going to move into the cells, and then it's going to, the cells are going to become more firm. The next thing we need is an isotonic solution. And for an isotonic solution, then it's 0.9 NaCl, and that means that it's going to have the same solute concentration as the inside of your cells. So when you go and get an IV or something like that, or any type of injection, they try to have it isotonic. Otherwise, if it's hypertonic, it's going to make your blood cells shrink up if they put it in your blood. If it's hypotonic, they'll make your blood cells swell up. And since we don't have cell membranes like plant cells, then our blood cells will just burst. If you try to make a microscope slide of red blood cells in water, you won't see anything in it because by the time you look at the slide, all the red blood cells have already burst. So we're going to do the 0.9% NaCl. Let's see. So here we have our purified water and a container. We're going to add 4.5 grams of NaCl. Take this, it's turned off, so I'll turn it back on. Make sure it reads 0, which it does now. And only 4.5 grams, that is a lot less than we had before. 2.83, 3.17, 3.3, 4.33. Oh, 4.50 exactly. So we got 4.5 grams. I wonder if you could even see that. No, it all just looks like 8 on there. Well, it says 4.5 for me. So I'm going to take this. You know what? This probably isn't going to displace any water at all, honestly. So we're going to go. Doesn't look like that's going to have enough water either. So I'm going to have to go get more water. I may actually use some of this because this was just the plain water. Okay. So we're going to go 50. 100. So there's something you normally never pour any type of solvent back into the original container. And I know I've done this twice, but I'm using all this water tonight. 150. 200. 250. And you know, I just wanted to compare to make sure I didn't lose count. And this one does have a little less than that one. So you can tell. And now we're going to put the salt in it. Salt now. And mix it up. And this one you won't need to heat up at all. It'll dissolve all the stuff. Real easy, I'm sure. And I'm going to use another one because the stronger salt water in the other one will make this end up being saltier than I want it to. So now we'll just, I don't remember how I poured that into here before. Oh, okay. I know what I did wrong. Put this all back. Actually, this pours better than I thought out of this, but still be easier to pour in a bottle. Okay. Yeah, 50. 100. 150. 200. By the way, I wouldn't put your funnel in there like I have it right now. 250, because it makes it easier to splash it. So we had 250 and we just have this very little bit. It's like 5 milliliters of water. So the rest, we're going to fill this up to the 50 line and make it 300. But see, that would have been 5 milliliters different. So the percentage of a solution would be like a 5% would be 5 grams per 100 mils. So, and it's 100 mils of solution, not 100 mils of water, 100 mils of solution total. Away. And now we're going to put this back in here. Normally I would have labeled this first, but these water bottles are so flimsy. You know, I should have written directly on them actually. So here is our 0.9% NaCl. 0.9% NaCl. Or my NaCl looks messy, but there you go, NaCl. And one thing that you should always do when you make a solution is put your initials, ABV, and put the date, and it's 3-22-23. Make sure you put the year also, because yeah, some people when they work in labs, they'll just leave something sitting around that's 100 years old, and you need to know that. (22, 23) and 3, 22, 23. I didn't write my initials on it. I didn't make this water. Okay. So those are our solutions made. Let's see, we have our water. We have our 0.9% NaCl solution, the isotonic solution, and we have the 300 milliliters of 15% NaCl solution. So those are our 3 solutions. The next thing we're going to have to do is the eggs without the shells. So we have to prepare the eggs without the shells. So I'm going to go ahead and move the stuff I used out of the way and move everything that I need for the eggs and put them in the right place. All of the eggs are going to be prepared the same. Okay. And if you want to have 3 eggs. So what you're going to do is, well, get some gloves. Right here. And safety glasses. And if you have glasses and side shields, you can do that because vinegar is acetic acid. And there's my gloves. So we're going to put each egg in a cup or glass and then cover each egg with white vinegar. And then we're going to cover each glass with saran wrap. Now, normally I probably would just put these in some stronger glasses that I have at home, but tonight I'm putting in these so you can see them. So I know eggs are very expensive these days. And actually though, Walmart has these really big boxes with like 60 eggs in it that are $12 a dozen. So that's 5 dozen for $12. That's where I got these. You may have 3 or 4 cracked. Is that 5 dozen? That's a lot of eggs though, anyway, in that big box. Even if you have 3 or 4 cracked and you only got a dozen out of it, that would still be a lot less expensive. So we're going to treat all 3 eggs the same. And we're just going to pour vinegar in them and try to get the same amount of vinegar. You can measure it out if you want to, because you want to try to do everything exact. But basically, like you wouldn't have one way up to here and another one like have like not even cover the egg. So here's our eggs. And they're covered in vinegar. This one needs a little bit more to be even, and now they are even. It doesn't have to be exactly even, but you want to get it close. So here are our 3 eggs in vinegar. And what's going to happen is, well, it's going to take about 3 days for your vinegar to dissolve the eggshells, or 2 to 3 days. So what I'm going to do is come back tomorrow and I'm going to dump out this dirty vinegar and then I'm going to put in clean vinegar. And hopefully then the next day there won't be shells anymore. And I'll be posting another video too of the eggs without shells and getting them out of here and so forth. But here's what I'm going to do now. Now you're going to cover them with saran wrap so they don't get all stinky and put them out of the way. If you have, you know, kids or pets, put them out of the way of your kids or pets. You don't want them eating raw eggs. So yeah, pour out the old vinegar, add new vinegar, and then once it's been, once it's done, then you're, you know, once the egg is removed, you're ready for this experiment. Now it has here in the lab sheet, it says about an hour before the experiment, place the eggs in a container of 0.9 NaCl. So the only reason I have you do that, you don't really have to because they're all in the same thing. So you don't really have to if you don't want to, but it starts them all at the same place, if that makes any sense. So that way, when you put it in the isotonic solution, it shouldn't gain any weight, but this is going to actually be in a hypertonic solution for a while because you have the vinegar dissolving calcium carbonate of the shell, and you're going to have all of that floating around in there. So you might want to start them all off at in an isotonic solution. Oh, look, there's all this introduction material that explains a lot of this. So this is really going to help. So we did the lab prep first. Okay, and I'm going to share this again. So we got everything ready. Then we prepared our solutions, and then we prepared the eggs, okay? And that's the part we're going to make sure we do tonight. Then we have the diffusion and osmosis lab. So here is your background material. So this will help you understand about diffusion and osmosis and what's hypertonic, hypotonic, and isotonic solutions. And then also about movement of water, cohesion and adhesion, and capillary action. So you're going to want to look at that. So for the diffusion, This right here, we'll do a little of this. We might as well. Might as well do what we can. So I'll stop the share. Oh, and remember, I'm going to cover these with saran wrap up your whole house. And I'm going to put those off to the side where dogs and pets, children and pets can't get them. I have little dogs, they can't get up here, but I'm still going to cover with saran wrap. So the one thing about diffusion is movement of molecules from an area of high concentration to an area of lower concentration. So this is just a demonstration of diffusion. And, you know, if you think of diffusion, if you put perfume maybe on your wrist or your neck, you have the high concentration of perfume on your wrist and on your neck, and the molecules diffuse from high concentration out to the air to lower concentration. But also when you make Kool-Aid, if you don't stir it, it's just diffusion. Now this water, you normally want to get a bigger container than this, and you want to get have it settle for a while, and then you can just pour in a drop of your food coloring and watch how it's just moving. And at first it's falling around, but you know, if we sat and we left this, then all of the blue food coloring is going to move from high concentration of the food coloring into the water area to lower concentration. And then the water is going to move from high water concentration to lower water concentration into the dye. And the molecules will keep moving until the concentration is even throughout. So that's going to be just even throughout. Now, if I move it at all, it's going to stir it. So, and that's going to probably take pretty quick, but I shake the desk a lot too. So if you did this with a big, like giant vase, and then you set it like on a back counter, but with a background that you could see behind it, and then you had it sit, then probably in 20 minutes it might be done. So anyway, this one started at 7:04 and it's 7:05, and we'll make our observations. But again, this is going to be moved. So osmosis in potatoes. You get 3 cups, and here's 2, 3. Can't move that. You can't move it. Okay. So for your osmosis in potatoes, let's see. Just want to try to move this. Don't move it though. Yeah. Oh, look, and it's 7:06. It's been 2 minutes. It's still not fully mixed. So here are our 3 cups. And you're going to want to label 1 distilled water, 1 15% NaCl, and then 1 is going to be 0.9% NaCl. Oh no, look, I put 0.9% water on here. You shouldn't, it should be 0.9% NaCl. So make sure that you change that on your sheet. 0.9% NaCl. That's at B, osmosis in potato, and number 2. Okay. So DH2O. 15% NaCl, 0.9% NaCl. Okay, so there are cups. And now what we're going to do is cut 4 potato slices about, ooh, and that's a slice, 4 potato slices about 6 milliliters thick. So you need a cutting board and a knife. And potatoes. I try to get all the slices of the same potato just to make sure that they are going to be, you know, similar. That other one's a little bit bendy, sort of, or you know, it's so we need 3 potato slices about the same size, and I'm just going to cut off the peeling. You could peel it if you want. Maybe you get more potato out of it. Let's see. Yeah, because I have still the peel on some of the corners. I don't want that, but so 3 slices, and let's see, there's 1. There's 1, 2, 3. This 3 from the middle is what I'm probably going to use because they don't have the bent corners. Okay. So we have our potato slices. And you know what? If you did this in the kitchen and cut your slices and threw them into Ziploc baggies, and then brought them in here for your lab, then that would be a way that you wouldn't have to, I don't know, waste the rest of the potato. But don't bring anything from the lab back into the kitchen. And don't use lab supplies over in the kitchen, unless it's a designated lab area. So we have our 4 potato slices about 6 millimeters thick. The peeling's not on them. We're going to weigh the potato slices, and I'm going to weigh it and put the weight next to the cup that it's in. So where did I put away that scale? Right here. Make sure it reads 0. And, oh, you know what? I don't even need a weigh boat if I don't want it, because I'm going to be putting it directly in that cup. I could just take the cup and make that zeroed out. That cup is zeroed out. The one in the distilled water is 13.69 grams. So that's distilled water, 13.69 grams. Okay. And sometimes I like to write it just in case. And then our next one. Okay, this right here is zeroed out now. And here, that's the 15%. I don't know which one it's asking for next. But this is 13.05. So I guess I skipped one and I'm going up here. That's 13.05. That's, wait a minute. Both of these say 0.9. Okay. Well, I'm going to change the third one to 15%. Okay. And then that one is 13.05 grams. 13.05 grams. Okay. And 13.05 grams. And then the next one is the 0.9% NaCl. These cups are very slightly different, so I have to zero it out, but not much. And here's the potato slice. That's 13.58 grams, 13.58 grams, and 13.58 grams. So we have our potatoes. I'll turn off my scale, put it away. Apparently this doesn't want to, my computer doesn't want to continue having a video. But here everything is even. Okay. Our 3 potatoes in the 3 empty containers. Now, what you're going to do is you're going to add solution, and it's not saying right now how much solution. 1 cup, the 2nd cup. Oh, you know what? I don't think I even say it says into the cup of 0.9 NaCl. I definitely need to make a few changes on this. So I'm going to go ahead and get a measuring cup. And then I'm going to add that in with that, so I won't have to use the, well, maybe I will use a graduated cylinder. Okay, and then see how much. The graduated cylinder. And I'm going to rinse it out with some not quite distilled water. And then I'll rinse it out 1 more time. My eggs, by the way, have bubbles all over them. That's because gases are forming from the breakdown of the shell. And the acetic acid must be CO2. So here we have this right here. And I'm going to see how much water are we going to put. We want to cover the potato pretty good. And there's 50. You know what? I am guessing since we made 300 milliliters, maybe some for the eggs, this could have got by with 50, but I'm going to go ahead and put the distilled water. I put 100 milliliters, and that's plenty. Look at that. That's plenty. So then I'm going to go ahead and use the 0.9% next, because our next week. And we're going to go 50. 100. 0.9%. So we have 200 left of the egg. And then we're going to do 15%. And we're going to do 50. 100. Now, I want you to notice something about these. Do you see something interesting? Oh, wait, real quick, the time. 7:16. And by the way, my solution over there is having diffusion. I don't think you can tell. It's mixed a lot, but it's still not completely mixed. But it's 7. It was 7:16 when we put that in here. 7:16 and 7:16. That means if we let this go for 45 minutes, it's going to be 8:01. So 8:01 is whenever 8:01, 8:01. Okay, so we're going to wait 45 minutes and take these out. But I want you to notice here, do you see something about the potato slices? But notice that it's sunken in the distilled water. The potato sunk to the bottom of the 0.9% NaCl. But look, the potato's floating in the 15% NaCl. That's because the 15% NaCl is more dense than the potato, and so it floats. So we're going to look back at these in 45 minutes, though, and we're going to see what's going to happen then. Now, this says fuel the fourth potato, and we find, this is why we have 4 slices. So that other slice that I didn't use, that was about the same size, okay, you want to feel it. Is it flimsy? This was an older potato. Okay, try to get a fresher potato than I have. Okay, because it had like potato eyes growing on it and all that. So mine has a little bit of give. Normally they don't have that much give as this one. And you know, feel if it's slimy or not slimy, and you can take off your gloves for this part, okay? But so things like that. Now we have 45 minutes to wait. But one other thing that we might do is the celery or Napa cabbage. So I'm going to pause this recording, and then I'm going to, you'll see some of this magically not appear and appear. So I guess my potato is a little bit not rigid, and it's, you know, I feel this, I can't tell with these gloves. Not really slimy. Okay, there. I needed to see if it was slimy. So we're gonna wait, and I'm gonna come back with some more supplies. Okay, now we're working on the third part of the lab, uptake of water in celery or Napa cabbage. And let's see. So it's going to be uptake of water in celery and or Napa cabbage. So right now we're skipping C because we're going to wait till our eggs don't have any shells on them, okay? And be careful handling them when you get to them. You don't want to break them. They're not super delicate, but still. Sometimes one will just break in the cup, so you never know. But right here, okay. So usually it's celery or Napa cabbage. I've got romaine lettuce tonight because I don't like celery and I always buy it. And it's like so much of it just goes thrown away. And then Napa cabbage, usually what I'll do is I'll make a stir fry with the Napa cabbage. I like it like that and it works really good. But I thought, why not try this romaine, the romaine hearts? I don't know why they wouldn't work. And it's something that I usually always have on hand. And remember to take pictures of this for your answer sheet. So what we're going to do is we're going to cut off the end of a stalk of celery or the bottom leaf of Napa cabbage. I mentioned to have a knife that's not serrated, and that's for this one because you don't want a raggedy edge. And I just broke these off, but now that it's cut, and I can actually see the vascular bundles. So vascular tissue in plants is what brings the water from the roots up to the rest of the plant. And it brings the products of photosynthesis, which are the carbohydrates, down from the leaves to the rest of the plant. So just like we have blood vessels in us that carry nutrients and waste and oxygen and CO2, these have, and that's our vascular tissue, their vascular tissue, like little bitty xylem and phloem, tiny little, like capillaries, little tubes. So here we have, they're cut from the bottom. Let's see. Place the celery or Napa cabbage cup side down into the cup of water in food coloring from part 1 of the lab. Oh, look, we have this already. I don't need to get any more. But I'll tell you, one thing I wish that I would have had is more food coloring. And by the way, the food coloring was pretty even at that point. I should have been watching it more, but you don't have to use this cup of food coloring either. Any cup of food coloring. If you want to change colors, you can do that. So I've found the two colors that don't work well are yellow and green, because they'll just mix in with the plant. And I'm going to put a lot more food coloring. And I'm just going to go ahead and put one of them in there. And it ought to go like up from the bottom through the vascular bundles at the bottom. It should go up through the leaf veins and out. You know how when you see like a carnation that has blue in it, then this is like how they usually do that. Now, I would actually like to use a different color, and I don't know if this is going to work. Too bad I used all of my tall cups that came with the lab kit. Let's see. It may work once there's water in it. I got more water. But I didn't. There's plenty more where that comes from. Oh, I have a purple. I'm going to try a purple. I've never had a purple food coloring. And I'm not going to be shy about how much food coloring I put in there. And then here's the purple. And then, you know what? I have a pink. So I'm going to get more water and another cup, and I'm going to do one in a pink. You only have to do one of these, I believe. Yep. And celery works really good. Napa cabbage works really good. And for this one, you know, I would leave it overnight, okay? Because you're going to be able to see a lot better if you leave it overnight. You can wait an hour and look at it and slice down here, and you'll be able to see the vascular bundles when you slice it. But what I would do is I would leave it overnight. Oh. And one thing I wanted to show you real quick is you're going to have to submit pictures of it. So like the celery, it really does look like this. And this is with a dark blue. And this one, it's so neat because these are the vascular bundles, xylem right here, xylem tubes, and that the xylem brings the water from the roots up to the rest of the plant, whereas phloem brings the food. Even though it starts with pH, it sounds like an F. Phloem brings the food from the leaves down to the rest of the plant. So it's the xylem tubes that you're seeing, and that's the stringy part of the celery that gets stuck in your teeth. So I hear. I can't even stand the smell of it. But so you're going to see the xylem, the vascular bundles and the xylem tubes are going to become filled up or be dyed, and even it'll go out to the leaves. So you'll be able to see some results if you wait like an hour, half hour, but you'll be able to see better results if you wait overnight and then look at it. Maybe look at it both times. So what I'm going to do is I'm going to leave this sitting here. I'm going to pause the Zoom, and I'm going to come back at 8, even though that's after normally we'd be at lab. I'm going to come back at 8, and it will have been 45 minutes, and I'll weigh my potato slices. So if you have any questions about any of this, feel free to email me as well. Okay, it's now been 45 minutes since we put the potatoes into the solutions. Okay, so now we're going to go back and look at the potatoes. And that is, let's see, share screen, diffusion. Okay. So 45 minutes, we did that. We felt the fourth potato, and we looked at its texture and observations and recorded them below. And now we're going to take all 3 out, but we're going to weigh them before we fill them anyway. So I'm going to stop sharing this because you'll have your own sheet out, I'm sure. With me, I find it easier, unless you have 2 screens and you have the sheet on the one screen, I find it easier just to print it out. Okay, so here we are, and here's our potatoes. And let's see. After your first potato slice has been in the distilled water for 45 minutes, then weigh the potato and then set it aside. Now, here's the deal. Oh, I should have brought a fork for this. That would have been nice, wouldn't it, to have a fork? Get a fork or a spoon. And here's my water. That way you won't have to dip in the water. And here's my potato. Now, if you dry it, don't squeeze the water out of it. That will wreck your results. Just pat it dry, flip it, and then that's 0. So I'm going to put it on the scale, 14.25. 14.25. So before it was 13.69 grams. Now it is 14.25 grams. Okay, so I'm going to set this next to the distilled water cup. Okay, the next one, the 0.9%. Okay, and again, a nice spoon or fork would be better. So we'll take it and just don't squeeze it dry. This still is reading, my scale is reading 0. You can see the scale. I'm putting it on the scale. 13.20. 13.20 grams. And write down the time. And now I'm going to look. Before this was 13.58. So it did lose a little weight. But there you go. And that one is the 0.09. And then for the last one, remember this before, how it was literally floating, and now it's sunk, okay? Which means you know something's changed, because even it's standing on end, but it still is not, you know, not floating. So we'll take that out. And again, just take it and don't squeeze any of the water out. Just flip it a little and make sure it looks like when you cut it. Make sure that's zeroed out. That's 12.15 grams. 12.15 grams. And 805. Okay. So there we have our 3 potato slices. And I'm going to have to clean this up later on. Now you want to feel the textures, okay? This one was in distilled water. Look at that. It doesn't hardly bend at all. It feels really like stiff. It's, you know, I can like stick my fingernails in it and it's like crunchy and stiff and crunchy and stiff. So water moved into this, by the way. You can tell because it gained weight. This one went from 13.69 to 14.25. So let's see the 1.9% NaCl. It's more flexible. It's not slimy or anything, but it's more flexible than the other one. So I'm going to go more flexible than, and I'm just going to put arrow up to the next one so I know. And then the one at 15%. Oh, look, that's even more flexible. And it does feel a little bit slimy. Feels a little slimier. It's more flexible than either of them. So I'm going to put most flexible. A little slimy. Okay, then we're going to go up. I know that there is a data table and it's on the answer sheet. So we're going to share the screen. And what you're going to turn in is the answer sheet, okay? So go to the answer sheet, and there is literally a table to fill in, okay? The initial mass of the first potato in distilled water, let's see, initial mass was 13.69 grams. And in the 0.9%, it was 13.58 grams. And then in the NaCl, it's 13.05 grams. Now, usually I get my potatoes closer. I usually cut them pretty close. These two are sort of close, but that one's a little far off. If you notice one's way off from the other, I would just try to get them as close as you can. They don't have to be exact, but okay. And then you have the initial potato texture. They were all stiff. They were all the same. I'm in solution. Final mass of the potato, this was 14.25, and it was grams. If I had had grams in the top here, I wouldn't have to write it here. And then the next one, not that it's that hard to put a G there, 13.20. And then the next one is 12.15. Okay, and then the final texture, we had crunchy and stiff. And then we had more. And then we had most flexible, slimy. Okay. And then the water, you know, this would be nice if it didn't have to go so much, but okay. There. Net movement of water. Okay, this one lost water, so water moved out. I wonder. And then this one, yeah, water moved out of the potato. And this one, wait a minute. I'm sorry, water moved into the potato here, not out. Okay, water moved out. And then water moved out. Oh, out of the potato. There. Now, tonicity. You know what though? This isn't good because we can't just have that net movement. That's not right. I'm going to go into, and then I'm going to go and use my calculator. Okay, because we need to have the whole movement of the water. We can't just say it moved out. I wish I did have one that moved out. So what we're going to do is we're going to go 14.25 minus 13.69, and we're going to say minus 0.56 grams. Okay, and this actually, I think I'll just go with this into, and it's not minus, actually, it's plus. And I'm just going to go ahead and put a plus. Oh no, we know water moved into, out. And then this is going to be clear, 13.58 minus 13.20 equals 0.38, and that's minus 0.38. That's actually a decent amount. And then out. And then we're going to go 13.05 minus 12.15. And that is minus 0.9 grams. So this one is it's more than twice the amount moved out. So you know that. And the 0.9% should have been isotonic, and it usually will be isotonic. Usually it looks really good. So I don't know why it's a little bit off tonight. I know the potatoes old, but if anything, I would have thought it would have gained. So the acidity is hypotonic for the distilled water. Maybe it's because I didn't use actual distilled water though. And you know that because you have a lower solute concentration in the water. So the water's hypotonic. And that's why water moves then from high water concentration, so lower solute but higher water, into the lower water concentration. It moves into the potato. The next one, that is supposed to be isotonic. You never, though, you always have to put exactly what happened. So I'm going to go appeared hypertonic. And then this one right here is going to be hypertonic. Yeah, I should actually have another column here where you can actually add it. Because at the final mass, it should be right here, gain or loss of water. But there we have it. So that is that one. And one other thing we're going to look at. Let's see, new share. We're going to look at the answer sheet. And there's one last thing on the answer sheet. Does it? Yeah, and it's going to be the celery or Napa cabbage. And we're just going to go ahead and look at that now. So I'm going to stop the share. And now we have this one. And now I'm done with all these. Oh, too bad I already got rid of those ones. And like I said, these next ones, you're going to want to actually do these the next day because you'll be able to see them all the way up. And I know it works really great with celery. It works good with Napa cabbage. So now we're trying the main. And I'm just going to look. And this one I did really quick. And okay, it looked like it had a crack. Doesn't really. So I'm just going to look and see how far it moves now because I can actually see. I can see the pink. I don't know if you can see that. It doesn't look like it, but maybe you can, but I can see it running up here. So it gets up to about here right now. Now here, you can barely see it, but I can see it. I don't know if you can see it at all. It's blurry. No, I don't know that you can see it. Oh, wait. There. You can see it, right? So those are the vascular bundles. You can see like 1, 2, 3, 4. And is there a fifth one in there? Yeah, it looks like it 5. So you can see it, and then you can see it starting to move up here. So I'm going to look at it tomorrow, and I might see a little more. And well, I will see a little more. This one, okay, this one's not going to work all the way because I didn't notice for some reason, but there's this crack right here. So let's see. This is why I always like to do these two. And I don't know what made this adjust. I think because I moved it before. Okay, you can sort of see it. You see what I mean? Now you can sort of see it. Really have to get back there. Focus just on this. Okay. Anyway, that one is going to look a lot better tomorrow. And this one, these are different food colorings than I usually use. Because I'm not the one who like gets all my supplies. This one is showing already though, and look how far it's showing it. You know, and this camera is just focusing on everything behind it. But look, and it's not even cut yet. So once I cut it, let's see what happens. I just cut as little as I could because, okay, now you can't see it as much, but I can see it. Maybe if there's nothing at all behind it. And it'll focus. Nope. No, and maybe if I make it focus on that. No. Oh, I had it for a second. Anyway, so you're going to be able to see how it's going to move up the xylem tubes, which are part of the vascular tissue of the plant. And it'll look a lot better in the morning. Oh, and one last thing, look at my eggs. It's already, they're already, that shows that are not on the plate. But you can see there's all kinds of bubbles. So you can see the vinegar, the acetic acid and the vinegar is reacting with the shell. And this one's already, I mean, they're already bigger. Oh, I shouldn't have done that there. Let's go wash up, but there they are though. So I'm going to come tomorrow and I wonder why these two are floating and this one's not. One of those eggs is not like the other. This is why I'm saying to take them and an hour before you use them, let them soak in the 0.9% NaCl. And that way they will, they'll all be starting at the same place. So if you have any questions about any of this, let me know. You can email me and I'll let you know if you have any questions, but that's basically it on how to set everything up. The only thing we didn't do were the eggs. And let's see real quick. Answer sheet, here it is. So when your eggs don't have any shell, then what you're going to do is you're going to get the 3 cups. Like, so I'm going to take my potato ones and clean them out, okay? And you're going to label them like we did for the potatoes. You're going to take the eggs out, and I like to use a tablespoon and take them out, put them on some soft paper towels, and gently dry the surface. And then you're going to weigh each one and make sure that you keep track of which egg you weigh that goes into each cup. And then you're going to like weigh it, record the weight, and put it in a cup of 0.9% NaCl and record the time. Then you're going to do the same thing and you're going to put it in the distilled water and record the time. And you're going to do the same thing for the 15%. If for some reason you only have 2 eggs, if one of them breaks, then don't do the 0.9% because that one shouldn't have any change. You should have more change with the distilled water and the 15%. But regardless, put them all in 0.9% NaCl, let them soak for an hour before you move them into the other ones. And after 45 minutes, if you want to go longer, you can go longer, but just make sure you write down the time. And here you're going to have the new weight, the times, and then remove. And then you're going to put your data in that table on the answer sheet, and you'll turn in the answer sheet. I believe there's some of them you take pictures of. So there you go. And that's it for tonight. Have a good night, and again, email me if you have questions. Goodbye.